Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello dear student, a very warm welcome to the TV learning programs. As usual, I'm your biology teacher Herbert, and today I will be still with biology class, or Form 3 class. We are still on the same unit, which is genetics. And today we are ending genetics. We talked about some of the genetic disorders. We talked about hemophilia. We talked about color blindness. So we are going to have a unit summary, and also going to have a testing of competence, or testing the understanding of the students, where we're going to do some of the exercise with me. So students, welcome again to TV learning programs, all right? So the lesson is having only one objective. It is to do exercise on the units, and we make a summary of the unit, and also we test the competence. When you talk about testing the competence, we are able to test the understanding to see if you have understood what I'm talking about. So student, are you ready with a pen and a book so that you can take the notes as well as doing the exercise with me? If you're ready, bravo. Let's go ahead and do the assignment, and also we make a summary. So if you're making a summary, we're going to see what you understand by the term genetics. We said genetics among the branches of biology, or sub-units of biology, that deals with the study of hereditary and variation. Last time we talked about variation. We talked about phenotypical variation and genotypical variation. So now we're going to have a unit summary. That is the summary we're going to have, a summary of what we discussed, all right? On genetics specifically. I'm talking about genetics, all right? So when you talk about genetics, is the study of heredity, the study of heredity. Or you can say is a branch of biology that deals with the study of inheritance and variation, all right? So inheritance, what is inheritance? Okay? Inheritance is the transmission of genetic information from one generation to another generation. Transfer of genetic information from one generation to another generation. What is that? We talk about chromosomes, thread-like structures that carries hereditary materials. Because on chromosomes, that's where we find the genes, all right? If you remember properly, if I talk about chromosomes, thread-like structures that are found in the nucleus of cells. They are thread-like structures. They are thread-like structures that are found in the nucleus of a cell. That is, in both plants and animals, I mean eukaryotes. Are you with me, student? So chromosomes are thread-like structures that carry hereditary materials. Hereditary materials, it means it carries genes. When you talk about genes, there are two definitions. Genes, unit of inheritance. Excellent. A unit of inheritance. Okay? It is a unit of inheritance. Or we talk about a part of DNA that encodes for a particular trait. Or a part of DNA that encodes. Talk about encodes. That is something not very new. Those are biological languages. The languages that are able to be understood by those who are doing biology, all right? That encodes for a particular trait. When I talk about a trait, some of you could think the teacher is talking about a trait. A trait is a character. Characteristics, the characteristics of being tall, characteristics of being short, and etc. They are made up of DNA, because when we talk about DNA, a storehouse for genetic information. And biology, we call it to be cellular library. A library of the cell, all right? So a gene is a unit of chromosomes that influences a particular characteristics. So genes are very important to influence a particular characteristics to appear in terms of the phenotype. So they are located on a specific portions known as what you call locus. A point or a location or a position where genes are located on chromosomes are referred to as locus or loci. Good, if you are with me. Two genes located on the same loci or on the homologous chromosomes form an allele. Student, they are words that we use what you call allele. Different versions of the same genes are called allele. Let me just use an example. If I have allele, I can have allele of being tall. Let's say have T capital and a T capital. These are allele. You can also have, they can just be different forms. All right? They can have T capital and a T small. Or we can have a T small and a T small. These are allele. These are allele. So we said a pair of allele influences the same characteristics. The allele that does not influence a characteristic in the presence of a dominant allele is said to be a recessive. Remember, I said dominant allele, they influence a characteristic to appear in phenotype. When you talk about phenotype, I mean external appearance. All right? Then that one that does not influence a character to appear is referred to as a recessive. Where the allele that influences a developing of a characteristics is said to be a dominant. That one that influences a characteristic to appear is referred to as a dominant allele. Good. Let's talk about homozygous. We are making a summary definition. We've talked allele. We've talked about dominant allele. We talked about recessive allele. Okay. We've talked about what else have you discussed? We've discussed about homozygous. We've talked about homozygous allele. We've talked about something like, yes, we've talked about chromosomes and etc. and etc. We are making a summary of the lesson. Homozygous. Homozygous refers to individuals that has two similar allele. Similar allele are called homozygous. Different allele, all right, different allele. This one is called homozygous. Okay. When they are same, when they are different, they are called heterozygous. Excellent. If you are with me, following me very well. So we said homozygous individuals has two similar allele. Okay? If I have, they are homozygous. Let's have examples. If I have B capital and a B capital, these are called homozygous allele. They can determine a particular trait. Are you with me, student? Good, if you are following me. But if they are different, if I have a B capital and a B small, a B capital and a B small, these are called heterozygous. All right? If I have A capital and A capital and A capital, these are homozygous. But if I have A capital and A small, these are called heterozygous. Good. Let's talk about monohybrid inheritance. The inheritance of a single character. Or that this is the inheritance that involves the study of the transmission of one pair of contrasting characteristics. The study of a single character is called monohybrid inheritance. Excellent, if you are with me. Good. Contrasting. Let me talk about, let me have an example. Let me give you an example of monohybrid inheritance. Monohybrid inheritance. The inheritance of a single character. It can be tall, the character being tall. Excellent. Tall. I'm talking about tall, that is monohybrid inheritance. Let me talk about tallness, and even shortness. Good. Shortness. And biology, sometimes we refer to as a graph. Are we on the same page? Are you following me? If you are following me, give you flowers to you if you are following me very well. That's good, all right? So this is a single character. We can have also color of flowers. Color, let me have color of flowers. The flowers can be red. We have red flowers, and we also have white flowers. Let me use a blue one. White flowers. This is monohybrid inheritance, the inheritance of a single character. We have also to do what we call test cross. Is used to determine the genotype. Used to determine the genotype of an individual with an unknown phenotype. When you're trying to know the genotype of an unknown, to know the genotype of an individual with an unknown phenotype, you don't know its phenotype. That one is referred as test cross. So what is test cross? Test cross refers to determine the genotype of an individual with an unknown phenotype. Excellent. Let's go through again. There's something that I talked about, Punnett square. Punnett square. What is a Punnett square? Is a diagram that is used to predict an outcome of a particular cross or a breeding experiment. Let me just use an example, if you are still following me very well. Example of a Punnett square. Punnett square method. Punnett square method. I'm making a summary. Okay? I will draw the graph. Okay? On that graph, I'm just maybe a cross between short and tall. Here, on that side, I will put here what you call gametes. Okay? The gametes could be female gametes and male gametes. Remember the signs that we do use. The female ones we use the signs like plus, then the male ones we use arrows. Okay? Are you following me? Gametes are going to be put on squares. All right? Here, tall, tall. Okay? And then you cross it with a draft. You cross it with a draft. Simple. Understanding that is a Punnett square. I said, what is a Punnett square? A Punnett square is a diagram that is used to predict an outcome of a particular cross or a breeding experiment. That one is called what you call a Punnett square. Here I would have T small and a T small. These are gametes. Remember, for the fact of life, gametes has to be circled. All right? Here, this one and this one is T capital and a T small, T capital and a T small, T capital and a T small, T capital and a T small. I was talking about in terms of tall and shortness. So the offspring produced here, they were all tall. Excellent, if you are with me. Now, then you have what to call incomplete dominance. Incomplete dominance. Not complete dominance. Incomplete dominance. One allele is completely dominating over the other allele, which is masked. You know what to call a mask? When you put a face mask, you are masking yourself. Nobody can recognize your face. Nobody can know that this person is this one and this one is this one. So the complete dominance is a condition where an allele is completely dominant over the other allele, which is masked and is expressed in homozygous state. It is expressed in homozygous state. Take an example. If I have T capital and a T capital, this is a complete dominance. The example that I've been doing there, it is a complete dominance. Excellent. Good. So we have another one which is called, and we said that one happens in a homozygous state. We have codominance. Both alleles are responsible for the transmission of a pair of contrasting characteristics, are dominant and are presented in a phenotype of the offsprings. What does it mean? It means that both alleles are fully expressed. That one is referred as codominance. Then we have codominance is exhibited in the Abo blood group system. All right? Blood group system, that's supposed to be blood groups where allele of antigen A and antigen B are dominant. Both of them, they are fully expressed. Are you following me? Let's talk about sex in human. Student, you remember I talked about sex in human. I said in human. How many chromosomes do we possess? We have 46 pair of chromosomes. And in biology, if there are 46, this is supposed to be 2n. These are diploid sets of chromosomes. Diploid sets of chromosomes. What is a diploid set of chromosomes? It has two complete sets of chromosomes. If it has two complete sets of chromosomes, that one is referred as a diploid set of chromosomes. Now, when it comes to haploid, this one makes half the number of chromosomes, which becomes to be 23, which is n. N refers to be haploid, half the number of chromosomes. Excellent, if you are with me. So in human, we have 46 chromosomes, which are diploid, but in gametes, we have 23 chromosomes. Out of that, 22 are called autosomes. They are called autosomes. Autosomes. If I talk about autosomes, they are responsible for making what you call body parts. Then the third one of the 23, this one is also what you call sex determination. Sex determination. Sex determination. If you talk about sex determination, this is supposed to be X and Y, because X and Y are the ones to determine the sex. For you to become a female or a male. Excellent. Good. So, sex in human is determined by a pair of chromosomes. That is, in female, it has 2 XX. In female, if you are on the same page with me, in female, let me just use here. In female, okay? What does it mean in female? We must be having 2XX. Then in male, we have X and Y. Excellent. That's what we said in human sex is determined by a pair of chromosomes. The female has 2XX chromosomes, while the male has an X chromosomes and a Y chromosome. Are we together? We talked about linkage that occurs where the genes are located on the same chromosomes, what you call sex-linked genes. These are genes that are located on sex chromosomes. The genes that are located on sex chromosomes are referred to as sex-linked chromosomes or sex-linked genes. Genes on the sex chromosomes are said to be sex-linked. They are linked on the sex chromosomes. All right? So that's what you call sex-linked genes. If you are with me, that's good. Let's talk about sex-linked genes are caused by sex-linked characteristics. Examples of sex-linked characteristics, we have homophilia and color blindness. When you talk about homophilia, that is sometimes referred as breeder's disease, a condition of breeding without stopping. Okay? We have color blindness, color confusion, red and green. You don't differentiate the two colors, so it can just be inherited. So the two sex-linked characteristics are homophilia and color blindness. All right. Color blindness and homophilia are linked to the X chromosomes, and is more common in males than in females. So it means that it is common in males than in females. Okay? Now, students, I've made a summary of the unit, but now I'm going to go through and give you questions where I will be giving the answers, and also we're able to work together these questions. Number one, state 2 types of variation. State 2 types of variation. 2 types of variation. Are you with me, student? Yes. 2 types of variation. State 2 types of variation. When I talk about 2 types of variation, one is continuous variation. If you talk about continuous variation and discontinuous variation. When I talk about continuous variation, this is a variation where there are slight differences among the organisms or among the species. And there is what you call intermediates. There is intermediates. When I talk about discontinuous, it has a clear-cut differences among organisms. Take an example, gender. Female and males are quite different. These are called what you call 2 types of variation. But under these 2 types of variation, they come from what you call genotypical variation. They are brought by the genes, which is called genotypical variation and even phenotypical variation. Number 2, give 2 sex-linked traits. Can you give me 2 sex-linked traits? The questions are there. We are working together the questions. Now, the 2 sex-linked traits that we've discussed homophilia and color blindness. These are 2 sex-linked traits. Number 3, which one of the following is not correct? I'm going to give you these. You tell me something which is not correct. Number one, inheritance of blood group involves both complete and incomplete dominance. I think this is correct because blood group can involve both complete and incomplete dominance. where blood can just be masked. That is incomplete, where that is complete dominance. Then incomplete dominance, that's where both of them can be fully expressed. Take an example, when blood group A has a recessive O, and blood group B can have a recessive blood group O. You will find a child with blood group O, which is incomplete dominance. Variation refers to differences within characteristics. Correct, 100%, it is correct. The last one, genes for normal color is dominant. Correct, 100%. Then the last one, genes located on homologous part of the chromosomes determine certain characteristics, which are referred to as sex-linked trait. That one is not correct. Let me have another question, which is very interesting. I will do that question, and now you're able to go through and do other questions, okay? There are a number of them that you're able to understand and able to do them very well, okay? So you're able to now to go through and do other questions. I've done almost like 3 questions. Now you're able to go through and do other questions that are following, okay? Excellent. Now, I've told you number 4. When a red-flowered plant of a certain species were crossed with a white-flowered plant, all plants produced were red-flowered. State the appropriate terms that can be used to describe that. The red-flowered plants, if all flowers that were produced were red, it means that one is referred as complete dominance. Complete dominance. The white flowers are recessive. The red flowers are dominant. Number 5. The allele for tall is a pea plants, Pisum sativum, which is dominant over the allele for the dwarfness. Using T to represent the allele for tallness, and T small to represent the allele for dwarfness. Write down the genotypes: homozygous dominant and heterozygous dominant. If you still remember properly, this is a simple work. We have what we call parental phenotype. Okay? We have now parental genotypes. What are they? Of course, this is supposed to be tall. You cross it with shortness, short pea plants. Okay? The gametes is supposed to be T capital and the T capital. You cross it with a T small and a T small. All right? Which is very simple. Here you have to form the gametes, which is T capital and the T capital. You circle the gametes for the fact of life. This one will form T small and a T small. Cross circle them. Okay? Very simple. T capital and a T small, T capital and a T small, T capital and a T small, T capital and a T small. Okay? Which is very simple. So homozygous dominant, they are tall peas. Then heterozygous, these are heterozygous pea plants. All right? We have another question which is there, homozygous recessive. You have to go through and do that one, which is very simple. So you also determine the tallness and even the shortness. I have another one, a pure breeding black mouse. Mouse was mated with a pure breeding brown female mouse. All offspring produced were black. Simple. We use the symbols. If I have, this is black, you cross it with brown one. Simple exercise. These were, they are represented by B capital. This is a B capital and a B capital. Okay? Then you are crossing with a brown, which is a B small and a B small. Of course, this one formation of gamete, B capital and a B capital, and then a B small and a B small, a B small. You circle them, all right? From here, you get a half of this, and you get a half of this. A half of this, and a half of this. Of course, this is B capital and a B small, B capital and a B small, B capital and a B small, B capital and a B small. So all offspring produced were black, as they are telling me. Okay? Now, using the letter B, now after from there, work out the genotype when you cross a heterozygous with a brown. If it is heterozygous, it means that I will get a B capital and a B small, then I cross it with a brown one. What will happen? You are able now to go through and determine you cross this one, which is very simple. These are going to form B capital and a B small. Then you have to form a B capital and a B small. circle the gametes, okay? Which is very simple. Simple exercise, simple mathematics, and simple to understand. This is a B capital and a B capital. Sorry, I said this is supposed to be brown. This is a B small and a B small. This is a B capital and a B small. This is a B capital and a B small. A B small and a B small, B small and a B small. Simple understanding. These are black, heterozygous black. These are pure breeding brown mice. So you can determine the genotype. Genotype, in terms of the genotype, I have two which are black. I have two which are, that is the phenotype. Okay? Two which are brown, in terms of brown mice. Then when it comes to the genotype, which is very simple, you are able to understand I have two B capital and a B small, and a B small and a B small, two which are B small and a B small. So student, that is a simple exercise you have to go through. So I can't leave without testing your understanding. That is the question you have to do. And in the next session, I will be able to give you the correct answers. Thank you very much for your active participation and listening. Enjoy the rest of the day and see you next time. Bye.